back to Gurukul CEC lectures. I am Dr. Kumar Shantanu, Associate Professor of Botany from Deshbandhu College, University of Delhi. We have been discussing various introductory aspects of Gymnosperms. We have till now covered some of the interesting features of Gymnosperms, their common origin, and their certain aspects of their identifying features. We shall continue our discussion by talking about characteristic features of Gymnosperms. The plants of Gymnosperms are sporophytes, and they have true roots, true stem, and true leaves. Sporophytes happens to be large, perennial, with active cambium producing secondary xylem, and most of them lack vessels except ephedra, welwitschia, and netum. As far as sporangia is concerned, it is one structure which bears all the spores. It could be microspore or it could be megaspore. They are born on a structure called as sporophylls, and the cluster of these sporophylls are together form cones. All different types of cones, they are heterosporous, and the two kinds of spores produce male and female gametophytes separately. The species of Gymnosperms, they are always dioecious. That means one structure, one cone will always be microsporous or megasporous, and the other one will be separately microsporous or megasporous. No one single cone will have both the types of spores in it. You can see with the help of this diagram, with the picture very clearly, that a female cone is a broad, more floristic in nature, whereas the male cones, they happen to be small and they are born in multiple number and they are born in clusters. In case of Gymnosperms, the true seeds are found. That is, the single megaspore is retained inside a megasporangium, which after fertilization in turn gives rise to or forms the seed. This megasporangium is always enclosed by an integument, and post fertilization the young sporophyte is formed inside this megasporangium, and this young sporophyte is also termed as embryo, which is always retained inside the ovule. Here we can see that this is a cone scale, or you can say that megasporophyll. On this megasporophyll, immature seeds or unfertilized ovules are found. After fertilization, these ovules will bear embryos, and they are still intact on the megasporophylls, also called as cone scales. The cone scales become woody and hardened later on during the development. In case of Gymnosperms, the gametophytes are very, very small, and they are absolutely dependent on the sporophytic generation for all purposes, be it nutritional, be it transfer, or be it their physiology. The male gametophyte is represented by 3 to 4 cell structures called as pollens. These pollens are transferred with the help of air to the female cones, and there these pollen grains, which are 3 to 4 cell structures, they germinate, and they germinate with the help of pollen tube. This process is also called as siphanogamy. So for the first time ever during the course of evolution in plant world, siphanogamy has set in, and that enhances the chances of fertilization. These pollen tubes are the passage which carry these antherozoids towards the egg cell, and this pollination always takes place with the help of wind or also sometimes with the help of insects. So now for the first time ever, the pollinating agent has been, a new pollinating agent has been introduced, which are insects also. The ovules and seeds which are born, they are naked and they are absolutely uncovered as compared to the angiospermic seeds. This can be represented with the help of this diagram, where the unfertilized ovule, it contains a megaspore. This megaspore, or the nucellus, also termed as megasporangium, that bears a spore. The megaspore is approached by male gametophyte, which is represented by the pollen grains, and then after formation the embryo is formed inside this nucellus. Nucellus represents the megasporangium, and the function of megasporangium is to provide nutrition to this developing young sporophyte. The seed coat here is derived from integuments, and this entire seed has no further integuments, and that is why they are also called as naked seeds. And this naked seed is one of the unique characteristic feature of gymnosperms. In case of gymnosperms, the archegonium has a very small neck, a venter canal cell followed by egg in its center. Single fertilization event takes place, that is, one sperm is involved in the process of fertilization. And as we have already discussed, this is because of the formation of pollen tubes. Proembryo is always free nuclear, and the endosperm is haploid, which is formed before fertilization. And here you can see that the archegonia, in case of gymnosperms, they have a small neck canal cells 2 to 3, followed by venter canal cell, and in the center lies a central large cell egg. This entire archegonium resides embedded inside a cushion tissue. During the course of fertilization, neck canal cells dissolve and the anthrazoids reach chemotactically towards egg, and fertilization takes place. After fertilization, this young sporophyte is produced. This is a young male gametophyte, and here this is 3 to 4 cell structure of a male gametophyte and a pollen tube here forming, and inside this pollen tube 2 male gametes, they travel all the way towards egg cell. Now this formation of pollen tube ensured that the fertilization is taking place and it is happening in a more efficient manner. The gymnosperms include both large trees and woody shrubs also. So when we talk about the morphology, we will see a range of morphological features, a range of habit amongst gymnosperms. There are certain species of gymnosperms such as netum, which is climber or which is woody climber in nature. There are species like Stangeria, they are like Microzamia, which are rhizomatous. Most of the gymnosperms are evergreen, but a few species of Taxodium and Larix, they are deciduous in nature. There are a large number of genera which exhibit xerophytic characters also. Here there are certain pictures of gymnosperms showing various different types of habit. Here you can see a woody climber, that is Lina of netum, Macrozamia, which shows rhizomatous morphology, a deciduous plant of cedrus, and xerophytic habit of Welwitschia. The gymnosperms possess a taproot system. Some of the genera, these taproots, they show certain symbiotic association. Now, symbiotic association amongst the roots of gymnosperm is very common. One such symbiotic association is of the blue-green alga with the roots of cycas. Because of this symbiotic association, the roots develop a coralloid structure which is very unique, and the presence of these coralloid roots are indication of the mutual association or endosymbiotic association between cycas root and the blue-green alga. Whereas in case of pinus and many other conifers, mycorrhizal association is very common. This mycorrhizal association ensures that the absorption of minerals and especially the potassium is taken from the substratum. Now, this in the picture you can see a mycorrhizal roots, a coralloid root. Here the roots, they look like corals, and that is why the term coralloid has been given to such kind of roots. And here you can see the long roots and short roots, and each of these roots, they have a mycorrhizal association. They are generally found in conifers. Both of these type of symbiotic association, that is with blue-green alga and fungi respectively, they help in the nutrition of plants. In those roots where there is a symbiotic association with fungi, that is mycorrhizal roots, the mycelium of the fungus forms an enveloping sheath which is called as Hartig net. Here you can see diagrammatically that the cortical cells, they have a very profuse channel of mycelium and they form a very thick net-like structure. Since it was first discovered by the biologist Hartig, so the term Hartig net has been given. The aerial trunks are mostly branched, but we do have many species of gymnosperms which have unbranched stem. For example, unbranched stem is characteristic of cycads. Those places where the branches are, where the stem or the aerial trunks are branched, the shoots could be dimorphic. That is, they are of two different shapes. They can be a long shoot or they can be a dwarf shoot. Here in the diagram you can again see two types of shoots, the long shoot and then the dwarf shoots also. The long shoots have an apical bud which grows indefinitely and they bear scaly leaves, while dwarf shoots, they arise on the long shoot in the axil of scaly leaf and they have a limited growth. You can very well see in this diagram that a long shoot, they have scaly leaves. These scaly leaves also act as a protector to the dwarf shoots which are going to come out from these long shoots. And the leaves are also dimorphic. There are certain leaves which are scaly in nature, and then there are certain leaves which are long foliage, also termed as needles in case of conifers. The dwarf shoots, they bear leaves at their apices. So here there is a diagrammatic representation of a dwarf shoot, and you can see multiple leaves coming out from the apices of these dwarf shoots, and this entire structure of dwarf shoot bearing multiple leaves. It is termed as spur, and this is a very common feature of any of the conifers. Now let us talk about the morphology of leaf. As far as leaves are concerned, leaves show a very long range of variation in form and arrangement. The leaves may be large. In that case, they are called as megaphylls, or it could be small in size. In that case, they are called as microphylls. Both simple and compound leaves can occur, and the shape also ranges from pinnate or fern-like in cycads to needle-like in pinus. Here, there is an interesting diagrammatic representation of various types of leaf. When we talk about morphology of leaves, we will encounter almost every type of leaf which has been shown in this diagram. This diagram is also very interesting because it also shows the evolutionary aspect of the development of leaf. So during Paleozoic and late Paleozoic era, the leaves mostly were simple in nature. So the lamina were entire and intact, and there was a single midrib, and from this single midrib the lateral veins were arising. Now gradually, during the course of evolution, the margins gradually started becoming serrated, and from these serrated margins the indentation further reached up to the main midrib, and because of these developments, finally compound leaves have evolved. So compound leaves are nothing but a more evolved simple leaves. These leaves ensured better absorption of carbon dioxide because of the increased surface area. They also ensured that there is less wear and tear because of any wind. This also ensured more photosynthetic productivity. The leaves of ginkgo are fan shaped, and those of Neetum superficially resemble dicots. In case of Welwitschia, they are strap shaped and persist throughout the life of the plant. In Gymnosperms, the foliage leaves are usually evergreen, and they are protected by a thick cuticle. The thick cuticle ensures less transpiration and less loss of water due to evaporation. Here you can see two extremes of the leaf structure in Gymnosperm. The leaves of ginkgo, which is almost like a fan shaped, and then there is a leaf of Welwitschia which keeps on growing throughout their entire lifespan because of the presence of some meristematic tissue at the base of these leaves. So we can also sometimes say that Welwitschia has the longest leaf blade in the entire plant kingdom. Now, besides foliage leaves, scale leaves can also be present, and arrangement of leaves on the stem is highly variable amongst Gymnosperms. They are generally spirally arranged, but they could be arranged as whorls, as the case in Cedrus, or they could be opposite decussate, as the case is in Cupressaceae and Neetum. The various types of arrangement of leaf on the petiole has already been discussed in our previous lectures. The venation also ranges from a single veined in most genera to reticulate in case of Neetum, parallel venation in case of agathis and welwitschia, and even dichotomous veins in case of jingo. In most of the gymnosperms, the leaf have raceme canals which occur in all conifers except taxus, and the leaf has stomata which are usually sunken. So, the sunken stomata on the surface of leaf is one xerophytic character which is very commonly found in case of all the gymnosperms. Here there is a picture which represents various types of venation pattern. We have a palmate leaf, then parallel venation, and then pinnate leaf where there is a lateral venation. Then there are different types of shapes. There is a lanceolate shape, there is a reniform shape, there is a ovate shape. So, this is a compilation of all different types of features of leaf, and to our surprise, we will encounter almost all these different combinations amongst gymnosperms. Now, let us talk about the internal organization of plant body amongst gymnosperms. In young stem, that is the primary stem, we encounter the vascular bundles which are open, collateral, and dark. When the secondary growth takes place, and due to the activity of cambium, we can see the development of different other types of secondary tissues. The woods in case of gymnosperm are characterized by absence of vessels except netum, ephedra, and welwitschia. When we talk about vascular bundle, which is open, collateral, and dark, let us try to understand diagrammatically. An open vascular bundle has a cambium between xylem and phloem tissue. A collateral vascular bundle has xylem and phloem tissues in one radial line, and the arrangement is in a ring. In case of gymnosperm, the wood comprises tracheids and the parenchyma is referred to as soft wood, where the xylem fibers are absent, in contrast to the angiosperm which has xylem fibers and the wood is termed as hardwood. There are two types of woods in gymnosperms, also called as monoxylic wood and pycnoxylic wood. The phloem possesses albuminous cells and the companion cells are absent in gymnosperms. If we talk about monoxylic and pycnoxylic woods, depending upon the amount of wood parenchyma present in it and the size of the pith, the wood could be called as monoxylic and pycnoxylic. The pycnoxylic wood is compact and it has narrow xylem rays and contains small cortex and pith. On the other hand, monoxylic wood has lesser xylem and more parenchyma present in wider rays, larger pith and cortex. Pycnoxylic wood forms the main component of the trunk and hence such wood is commercially important. Monoxylic wood is not important commercially. You can see difference between monoxylic wood and pycnoxylic wood with the help of this diagram, where the tracheids are diffused, the parenchyma cells are much more in amount, whereas in case of pycnoxylic wood the parenchyma is very less, and the arrangement of these tracheids are very compact. Because of this compact arrangement of tracheids in pycnoxylic wood, they are commercially very useful. As far as the roots are concerned, in roots the xylem is diarch or it could be polyarch. The tracheids constitute the major part of the xylem. An important feature of the gymnosperm leaf anatomy is the presence of transfusion tissues also. When we talk about the anatomy of leaf, the leaf has an outer epidermis which is covered by cuticle, and the epidermis is interrupted by sunken stomata. In the center of the mesophyll tissues, which are green mesophyll tissues, lies the vascular bundle. Now, in these mesophyll tissues lies resin canals, which are characteristic feature of most of the conifers. The vascular bundle contains a very unique tissue which are called as transfusion tissues. As far as the vascular bundle is concerned, there are two different groups of xylem and phloem. Xylem is towards the inner side and phloem is towards the lower side. So this is all about the anatomy of leaf, stem and roots in case of gymnosperms. Now, in our following lecture, we will continue our discussion towards the reproductive aspects of gymnosperms, and then we shall try to understand the biology of gymnosperm by going through different species such as Cycas, Pinus, Netum, Anjingo, etc. Thank you so much for your patient listening. I hope I have been able to convey the various morphological and anatomical aspects of gymnosperm in much detail. Thanks a lot.